Hello everyone, and welcome to the third lecture of this second edition of the Current Topics in Heritage Science series of IPU-NHS Academy. This series is organized by emerging professionals in the frame of the IPU-NHS project and the European Research Infrastructure for Heritage Science. My name is Diego Quintero Balbás, and I am a researcher at the National Institute of Optics in Florence, and I'll be moderating today's lecture. Our topic today is chronological tools dating human evolutionary history. The recording of this lecture will be available later in the E-RIHS YouTube channel, and at the end of the lecture you will receive a survey. Please take a few seconds to let us know your opinion. And you will receive an attendance confirmation of this lecture after you complete the survey. You can ask questions at the end of the presentation through the Q&A function at the bottom bar of the Zoom window. And if you are experiencing any technical difficulties, you can contact us using the chat. So I am very happy to present today our speaker, Dr. Altug Hasozbek. He is a geochemist that is working at the U-Series lab, which is part of the CENIEH, the National Center of the Research on Human Evolution in Burgos, Spain. He is specialized in isotope geochemistry, in understanding geological and environmental process through isotope studies. His work primarily involves the use of instruments such as ICP-MS, MC-ICP-MS, and TIMS for radiometric dating. Thank you, Dr. Altug, for accepting and doing this presentation. Now, the floor is yours.
Thank you. Thank you. I will share my screen. I think it's visible now.
Yes.
All right. Okay, perfect. Good afternoon, everyone. And today I will try to talk about the chronological tools especially in dating human evolutionary history. And I will try to focus on some chronological tools. But before I get started, I would like to show this video. And this video is actually available in the Big History website, which is an open source for educators, for teachers, for professors, and for students as well. So this video is just going through all the universal time scales, and then to just show us the thresholds of what exactly happened during the history of our Earth, and as well as it goes back forward to our solar system. So I just want to start that with this video, because just to point out how important is just to fingerprint by using the time, and then link with the history of our Earth formation and our solar formation, and after especially our collective learning. So here you can see, go through that, just like from Quaternary period to now we are in the 66 million years in the dinosaurs time, and then you go through the Big Bang, and you can see that all these thresholds in a big scale, which is here is that in detail. There are some Big Bang, the stars light up. They're all fingerprinted, or I would say isotope printed by dating or by time. But there is one thing I would like to point out is when you look in detail that how the scale, when you get close to our time, our recent time, with the collective learning, is just that the events of the history is just spiked, it's just exploded. So today, in that sense, I will try to cover that absolute dating in human evolution research, why it is so important, And by using some chronological tools for absolute dating, I will try to give some frame of these techniques, especially paleomagnetism, OSL, and ESR. These three techniques is not especially my my area, but I'll try to I'll try to transfer the message from the researchers from the CENIEH by using their slides. But I will more focus on the uranium series dating used in the human evolution science. And then I'll try to conclude some CENIEH approach to utilizing absolute dating techniques and with some future aspects. So this is actually, I would like to show that's why we celebrate the birth age of the Earth. This is the zircon you are looking for. And this is by using the uranium-lead laser ablation technique, from these tiny zircon grains, we know the date of our Earth, so which is like 4.5 billion years. But from that time to here quaternary, things get a little bit complicated because it's just when you look to the quaternary period, and it's just like, I would say, like from 2.8 million years to current time, is our actually dating techniques is a bit limited. I mean, there are lots of techniques, but in archaeology, especially for the radiogenic isotopes, it gets a little bit complicated. In the coming slides, I will try to frame why it's getting complicated. So here is just like, especially in this, the second one, you can see some uncertainties of the techniques. For example, the luminescence, ESR techniques, depending on the lab, depending on your samples, of course, it varies, but there is always an uncertainty in these dates involved, so which we have to bear in our research and research questions. So by saying that, is actually, this is my take-home message. This is my conclusion of this lecture. It's just for the stratigraphy and dating, we use the chronostratigraphy. It's just, you can name it as isostratigraphy or seismostratigraphy, sequence stratigraphy, depending on your field. But in either stratigraphical tools, you have to calibrate your geological scale. What I mean that is just you have your stratigraphical section in a superposition based, the older rocks in the bottom, and then going to the top, you have your younger series, and you just like combine with this knowledge some recognition of some events, for example, a kind of a volcanic eruption or a fossil data. And when you combine this data, you include your radiometric dating, and you can calibrate your geological timescale, which you can say that, okay, I have an approach of absolute dating. So this is actually go-home message. Even though the absolute dating techniques have improved a lot, it's just very important to cross-checking with stratigraphical context remains very, very important. It's a concept of stratigraphy is fundamental to research in geology, in geochronology, but especially on human evolution. So let's start with the paleomagnetism. As I said at the beginning, that this is not my field, but these slides are provided from the Dr. Joseph Pares. He is the coordinator of geology and geochronology. Please feel free to ask any further questions or any requirements for this technique, but I'll try to share some brief info why paleomagnetism is important in archaeology and in human evolution. So basically, what is paleomagnetism? Let's start with this question, is the study of the record of the Earth's magnetic field observed in various geological and archaeological materials. So, and how it works is just actually works with the Earth's magnetic field, and that aligns with the iron-bearing minerals in rocks and as they form, or in your sequence. Over time, these minerals retain the magnetic signature, and then this magnetic field, that is the intensity of this magnetic field, is indicating that when the rock was formed. This is not an absolute dating technique. It's a way of saying that it's best. I think that this is perhaps best describing this technique is just as a crossing between the numerical and the relative dating. And in that sense, it provides the basis for a special kind of stratigraphy and probably the same kind of the dating implications as biostratigraphy. And it's also the foundation for this paleomagnetism are the past fluctuations in the intensity and the direction of the Earth's magnetic field. I am trying to say that is just, for example, from a anthropological perspective, is that the most important changes are in the polarity, and from times when a compass would point to north, and then times when it reverses and it will point to the south or vice versa. So this is the typical, it's just the columnar section, or is, I would say, the magnetic stratigraphy is for human evolution. With the red indicated is that the most important date is for the, by using this technique is the Brunhes and Motoyama boundary, which is an estimate of 780,000 years. Is actually, I will show why it's very important, especially on Atapuerca site, is just published from the CENIEH researchers. And these crons, what you see here is the crons and the subcrons have been dated from the paired radio potassium and paleomagnetic readings. They're all actually cross-calibrated with volcanic rocks and from paired climatic and paleomagnetic determinations in the deep sea environments. And there are prominent examples you can easily find in the literature, in the Atapuerca site, as I said, in Grandolina and the Cima del Elefante. These are the sites of the Atapuerca. These are the main caves names of the Atapuerca. I will give some examples in the coming slides. And these reversals in the current within the Earth's fluid is actually the core, almost certainly cause some shifts. So through these currents are poorly understood, but the timing of the shift appears irregular. So shifts do actually clearly occur in two scales. I would say it's just that the long periods characterized by essentially the same polarity and is punctuated by much shorter intervals of opposite polarity, and the long periods lasting hundreds of thousands or even millions of years are called polarity crons and formerly epochs. And the shorter intervals lasting no more than a few tens of thousands of years. So here you can see that they're known as the polarity subcrons and formerly events. So how do we sample and how to get your sample is actually the sampling and analyzing is a bit complicated because you have to be very careful. Even though you don't need that much tools to get your samples, you just need a hammer and then to extract your sample. But sometimes your sequence is a bit softer, it's easier, or it's just a rock. You need some drilling tools, things like that. But here I just want to show, actually, it's a summer training course. I will share some photos. Here you can see one of this student is trying to get sample for paleomag in the Atapuerca site. This is a bit softer level. So it's a little, you can see the little squares in the reddish colored zone. And it's just the same, the layer by pointing to north and is just taking the note of this north. And then after that, you collect these samples and analyzing the magnetic properties of these samples in the paleomag lab, and then the data interpretation comes afterwards. And this is the photo from our CENIEH, the paleomag lab. And this is the one of the best examples that was applied to date the Homo ancestors in the Grandolina site. Here you can see that by using the paleomag and by using the reverse and the other polarities, you can give the maximum date of your fossil. Of course, in the coming slides that I will also show you that it's very important to verify and cross-calibrate your data with other techniques as well. In the coming slides, I will also mention on that. This is another slide. It's showing that how it's applied. You can see the Brunhes boundary and the Motoyama boundary in the Grandolina site. So let's move on with the other technique, which is OSL, Optically Stimulated Luminescence. The coming slides are provided by Dr. Alicia Utande. She's the researcher at CENIEH and the OSL. She kindly provided me this information. And if you look to the description, this what is OSL is just a technique that to determine the last time mineral grains were exposed to sunlight and or sufficient heating. And the luminescence dating provides the time when the mineral grains were exposed to the light or high temperatures is over 400 degrees. And for the last time before being deposited, as just you can see in this illustrated graph. And sampling and analysis, which was also is during this Hyperion summer training here, as Dr. Alicia Utande showed how to take the samples. It's actually, sampling is one of the most important things for other techniques, but for OSL, it is you have to very carefully extract your samples from the sediments or artifacts without exposure to sunlight. So you have to make sure that when you send your sample to CENIEH or to other OSL laboratories, that your sample shouldn't even just exposure for seconds of the sunlight. And then the samples are prepared in a designated areas, light tight conditions, and the mineral grains are stimulated usually by light in a lab to release the stored energy and the light which is measured. And the amount of luminescence actually is used to calculate the time since the last exposure to this light or heat. Here you can see the tubes here is to be measured by the OSL technique. Here is the formula, general formula, is you can see you just basically your accumulated dose divided by the dose rate, and depending on the calculating the time since the last exposure to the light and heat. And this is the example from the Hyperion summer training. These are the students are trying to take samples from the sediments of Atapuerca. And this is the one of the recent published paper, which was dated by Dr. Alicia Utande, and it's the 2023 just published in Quaternary Science Reviews. This is the Neanderthal footprints in the Matalascaras trampled surface. It's the southwest of Spain, and there were some previous OSL dating, and now they just did some more OSL dating. Here you can see that the cross section and the blue ones are the dated zones by using the CENIEH OSL facility. And another example is Atapuerca Grandolina. This was done by Duval et al. This is also, as I mentioned before, in the PaleoMag. This is also just the blue ones are indicating the OSL techniques. And here you can see that is the bold indicated dates are the results that are interpreted as the most reliable age constraints along this sequence. And let's move on with the ESR dating, electron spin resonance. This is called ESR, and this also the slides and the information is provided by the Dr. Mathieu Duval. And he also gave a lecture here during the HS Hyperion training. These some of the slides and information from those slides. So when we look what is ESR dating is a radiometric dating is method used in archaeology and geology to date the crystalline materials by observing the behavior of electron charts trapped within the material. So this is effective over 2 million years. And it is the irradiation in this technique is depend on this observation that irradiation naturally occurring in the radioactive isotopes, mainly uranium, thorium, and radiopotassium, causes the electrons to accumulate in defects with the crystalline lattices. The aggregate numbers of the trapped electrons can be measured by spectrometers, and the rate of that which they accumulated can be estimated from the level background radioactivity which is a substance was exposed. And the number of the trapped electrons divided by their annual accumulation and rate then provides the last time in the crystalline traps were empty, and then ESR is applied mainly to basically dental enamel or where formation produced initially empty traps. So by saying that, this is another example from the Atapuerca. This is the first direct ESR dating of a hominin tooth from Atapuerca grandolina TD6. This supports that the antiquity of human ancestors, and it was previously dated by PaleoMag, and there are some additional data is used by ESR, and this is already in 2018 published by Duval et al. Quaternary geochronology. And so now I'm gonna go to actually my field. Some uranium series dating is overall is the uranium series dating is one of the other radioactive dating, which is now is also at CENIEH we are commonly using by also adding with other data provided with other ESR techniques or OSL techniques. So I'm just basically mention the basics, some technical approach and research approach. Here you can see just a summary of that. The left side is the PaleoMag dating and this is carbon 14, which is out of our scope right now. This is the uranium-thorium and luminescence. And here you can see that all the techniques can be applicable for the human evolution science. And here now we are going to focus on from that uranium series, which is a 1,000 to 500,000 years, the age range application of this technique. And mostly if your mineral has uranium bearing mineral, it's like coral, shells, teeth, you can easily use this technique by using the radioactive decay 234 to 230 thorium. So this is, we are using actually is this technique is a radiometric dating used in archaeology and geology. And this is effective depending on the lab and depending on the instrument. But because of the half-life of the 234 uranium, this dating technique is limited to more or less 500,000 years. And for example, in our case, in our lab, we can go to the oldest one we dated here is 480,000 years old. So as I said, this technique, we use the 234 uranium to 230 thorium decay. And here is just like to understand some basic concepts. There are some useful terminologies that worth to mention. As I said we call it uranium series, but in literature you can call it uranium-thorium series. Secular equilibrium, the disequilibrium dating, 232 to 234 uranium, and you will hear closed open systems and isochron methodology. So by saying that this is called a disequilibrium technique, is this when the fractionation event happened by the time one, and then it's eventually goes approach to the secular equilibrium in the decay series of this time. So this is actually defined by the Edwards diagram in 1988. So decayed behavior within this chain is the reactions to lead to use the uranium series disequilibrium as a chronometer. So the time scales of any dynamic processes which causes any fractionation can be quantified by using this disequilibrium. And so this is I'm not going to go through this detailed of the formula, but basically Ivanovich and Harmon formula is one of the common used. Important points is just like your sample shouldn't contain any initial 230 thorium, and depending on the uranium content of the sample, the age range, as I said, it can go to 2,000 to 600,000 years. And there is also a space that you can define your initial correction. So isochron's corrections technique can be used with the total sample dissolution, which is described in Ludwig 2003, and other approaches are available, which is a little bit in discussion. But let's look at how we do sample and analysis. Let's go through the sample preparation, chemical separation, and some multi-collector ICP-MS measurements. This is hard work actually. When you get a big spill time, you have to cut your sample. you have to polish, and then you can use a manual drill. Basically, you need around 50 milligrams of powdered carbonate samples. Here also, like if you have a very precious sample or you want to look in detail, you can do micro drilling. Here you can see in the video, this is a small example, which is like 0.8 millimeter radius, and you can follow, it's just like in laser ablation, you can sample your just carbonate powder in that detail by using the micro drill. So is this to use the solution-based dating, you need a clean lab, and CENIEH facilitates one of the unique clean lab here. It's a metal-free lab, and you have to first chemically separate your uranium and thorium. And this is described in the Portman et al. 2013 paper. You dissolve your sample, and then you have to separate your matrix, and then you have to recover uranium and thorium, and you measure your samples in the multi-collector ICP-MS. And this is our instrument here at CENIEH. We have 9 Faraday cups, 6 multi-ion counters, jet interface, and we have a Aridus 2 system, dry plasma system. And multi-collector ICP-MS is basically works is like this is an example of TIMS and the element single collector ICP-MS. And just when you have your solution, you can use the plasma power to focus on your isotope range, and then you can detect your samples in your software. So this is some of one of the example, and then you use the reduction script here. We are applying the Portman et al. Technique by using the Mathematica, and then we date. Here is an example, one of the uranium series dating. This is actually the one of the first time I'm using the image generated by the artificial intelligence, which gives an interesting image. But this is the speleothem dating, for example, uranium-thorium. This is from the Portugal dating. We dated, and this is another example that was dated here, the sample, the lion bone sample from Andalusia. And let's focus on real samples, real fieldwork samples. This is actually pre-Roman uranium-thorium datings just recently done here is of an aqueduct near ancient Lepidus Aegean region, Turkey. And this is an Hyperion project we perform here. And I think this paper is right now in review. Here you can see the Western Anatolia, the ancient cities. They found this aqueduct, and then from this aqueduct, they found this this formed sinter, let's say travertine, and then we dated this travertine, and this turns out this is a pre-Roman aqueduct sample from the Western Anatolia. And there are some other unique examples, which is from this one is from Hoffman et al. He applied this method to get uranium dating from the calcium carbonate crust associated with Paleolithic cave art and application to Iberian sites. So basically what I cover here is just like a CENIEH approach to absolute dating is paleomag, luminescence, electron, ESR, uranium series, and cosmogenic nuclides. I didn't have enough time to just cover the cosmogenic nuclides, but you can always access from our website, and you can reach Dr. Toshi to get some information for these techniques as well. And this is the CENIEH approach to absolute dating. Is actually we applied almost all the techniques to give an absolute date, because this is very important to make sure in an archaeological study. So here's a very good example from Duval et al. He applied uranium series, paleomagnetism, ESR, and OSL techniques to have all the levels of covered by the absolute dating technique. And so it's just like as a conclusion, I will say that different dating techniques exhibit limitations for sure. And the concept of stratigraphy is fundamental for the research, and different dating techniques are strongly encouraged to be used in human evolution. But just to, I know I don't have enough time, but just to cover up with what is next for that, I think this might be an interest, because there are some options, semi-destructive dating technique, for example, by using laser ablation coupled mass spectrometry, as well as non-destructive dating technique, which is called chemical dating, by using some advanced photon sources. I'm just gonna quickly talk about this. This is the uranium-thorium series dating limit. As I mentioned, this is the 450K, but just the missing chronological tie-ins, you can use uranium-lead dating, which gives you that, in that sense, that you can date older than 450,000 years to 300 million years. So by using that, you need a closed system. You have to pre-screen your dating. It is some of the labs are very well maintained in terms of clean labs, so they can apply this. But for the laser ablation, there are still some questions to lower the uncertainties of this technique. And here you can see that how the uranium-lead laser ablation can give you some more data, because this is on the left side. You can see the speleothem is, for example, this speleothem is from let's assume that this is from Atapuerca side, and you want to have this precise dating. You have to go through with the laser ablation like more than 100 samples. You don't need a chemistry to separate your uranium and lead, and you can actually have your Concordia, and you can have your data in, I would say, 60 seconds by using the laser ablation technique. And for chemical dating, this is also previously done. I did some laser ablation work, University of Miami. It's basically, I focus on strontium isotopes, but what I'm trying to indicate that this is ongoing process, so this is possible to use laser ablation technique to lower your destruction in your precious samples. And the last one I want to focus is the synchrotron dating, which is totally non-destructive. This is from the University of Chicago, advanced photon source, which I did my postdoc here. Especially, I was focusing on the uranium characterization, but I had the chance to use the advanced photon source. This is the unique photon LINAC technique that you can have a speed of light electrons provided by the LINAC and then boosted in the synchrotron. Here you can see, and then it's actually distributed to the point of interest of the sectors. And here I use the sector 2 IDD, and to see that without destroying my mineral, if I can chemically date my sample. This is my zircons, and I try to see that without destroying my samples, this nice XRF images with low energy and high energy. And at the end, chemically, I have the data, but unfortunately, I couldn't date this, especially this sample. But there is a way that there is a paper that they did, because they have a very high amount of uranium and thorium. So without destroying the sample, they managed to use this technique to have and it's called chemical dating. And I assume that it's going to be the future, and I will say that it's going to be very applicable for the archaeological sites. So here are my acknowledgements. Please, I just repeat, but I have to thank for paleomagnetism slides to Dr. Joseph, OSL slides to Dr. Alicia, ESR slides to Dr. Mattia, and some other colleagues for CENIEH, and some references for your interest. And this is a nice photo from our first Heritage Science Academic Training Camp from Atapuerca. And yeah, thanks for your attention. I think I'm over 1 minute and a half, 30 seconds. Thanks a lot.
Thank you so much, Dr. Altug. So just before going to the Q&A session, I'd like to present our next lecture. It's going to be on January 25th of next year. And it will be a lecture by Emma Carune, and she's going to talk about open and fair science with a case study from the fair phytoliths. Okay, thank you.